Hello everyone! Today we're going to answer the question, how does our nervous system develop? In other words, how do we go from a single cell to babies with fully developed brains? Naturally, this doesn't happen in a day. In fact, it takes months. Some neuroscientists would even argue years. It also occurs over a number of steps, and today we're going to go through those one by one. We start off as a single cell, known as a zygote. Then, the zygote undergoes a number of cell divisions, collectively known as cleavage. After this process, we have a morula, or a solid ball of cells. The morula undergoes a process known as blastulation to become a blastula, which is a hollow ball of cells. The blastula's fluid-filled cavity is known as the blastocoel. The entire blastula undergoes the process of gastrulation to form a gastrula, which is a three-layer ball of cells. Let's go through these three layers one at a time. The innermost layer is known as the endoderm. This gives rise to the inner linings of our digestive and respiratory tracts, including the lining of our lungs. Next up, we have the mesoderm, or the middle layer. This helps us get around in a number of ways, as it gives rise to our musculoskeletal system, our circulatory system, and our reproductive organs. Finally, we have the ectoderm, which is the outermost layer. It will eventually give rise to our skin, hair, nails, parts of our nose and mouth, the lens of our eyes, and most importantly for this video, the nervous system. In week 3, a portion of the ectoderm thickens and forms what's known as the neural plate. This neural plate then begins to fold inward in the center, forming what's called the neural groove. The neural groove consists of two folds, one on either side, as well as two peaks known as the neural crests. In week 4, this neural groove closes, forming a neural tube. This neural tube will go on to form the vast majority of our central nervous system. Its cavity will later develop into the brain's ventricles. If you're not sure what ventricles are, be sure to check out our meninges video on YouTube. To continue, the neural tube balloons to form three sections known as the three primary vesicles, the first of which is the prosencephalon. This is Greek for front brain, and this section will go on to form the forebrain. The term mesencephalon is Greek for midbrain, and this section will go on to form the upper portion of the brainstem, which is appropriately called the midbrain. Finally, we have the rhombencephalon. This section was named for its rhomboidal shape, and it will go on to form the two lower portions of the brainstem as well as the cerebellum. In week 5, we witnessed the subdivision of these three primary vesicles and the consequent formation of the 5 secondary vesicles. In other words, the prosencephalon and rhombencephalon will each subdivide, and the mesencephalon will stay the same. The prosencephalon will subdivide into the telencephalon and diencephalon. The telencephalon consists of the two cerebral hemispheres, while the diencephalon consists of the thalamus and hypothalamus. As mentioned earlier, the mesencephalon does not subdivide any further, but the rhombencephalon will. It will form the metencephalon and the myelencephalon. The metencephalon consists of the pons, the middle portion of the brainstem, and the cerebellum, while the myelencephalon consists of the medulla oblongata, the lowermost portion of the brainstem. Let's review some of those tricky terms. The midbrain, pons, and medulla oblongata are the three portions of the brainstem, and here they're depicted as they are in the brain from top to bottom. Naturally, nervous system development doesn't stop after 5 weeks, but rather it continues throughout pregnancy and even after birth. During months 3 to 5 of pregnancy, or during the second trimester, we witness a burst in neuronal production and neuronal migration. The burst in neural production makes sense, as we start out as single cells, but end up as babies with over 100 billion neurons. During neuronal migration, these cells migrate to specific locations in the brain, where they will eventually serve specific functions. After birth, we see continued formation of neural connections and the formation of myelin sheaths. If you're not sure what myelin sheaths are, be sure to check out our video titled Cellular Neuroscience on YouTube. But just to give you a recap here, myelin sheaths serve as an insulating layer for neurons, and they increase the speed of neuronal signals down the axon. That, my dears, is how we go from a single cell to babies with fully developed brains. That's all for today. Thanks for watching. Bye.